Every living cell in our body carries genetic instructions. Most of us are familiar with DNA inside the nucleus. This is nuclear DNA. But what many people don't realize is that the cells also carry a second genome hidden inside structures called as mitochondria. This second genome is mitochondrial DNA. It might be small, but it plays a very powerful role in keeping cells alive by producing energy. In part A, we will explore what mitochondrial DNA is, where it came from, and why cells evolved to maintain two genomes instead of one. What is extrachromosomal DNA? Traditionally, DNA is located within the nucleus. However, not all DNA is stored there. Some genetic material exists outside the nuclear chromosomes. This is known as extrachromosomal DNA. This idea tells us that the genetic control in cells is not centralized in one place, it is shared across different compartments. So this is the different extrachromosomal DNA that you can find outside nucleus. One is mitochondrial DNA, which is present inside mitochondria. This is there in all eukaryotic cells. And then there is the plasmid DNA, which is present inside the bacteria. It's important for it has all the antibiotic resistance genes. And then you have chloroplast DNA, which is inside chloroplast, which is present in plants, which helps in photosynthesis. And okay, so these are the three different types of extrachromosomal DNA that you can find. Okay, examples. So that's what. So there are several types of extrachromosomal DNA. You have mitochondrial DNA that is found in the mitochondria and supports energy production. It's found in all eukaryotic cells. Then there is chloroplast DNA, which is found in plants. It supports photosynthesis. And then we have finally plasmid DNA, which is present in bacteria. It carries genes that help survive. It has like antibiotic resistance genes. Together, these examples show that cells evolved by keeping a useful genetic system from different origins. So where did the mitochondria came from? So according to the endosymbiotic theory, mitochondria were once free-living bacteria. An ancient host engulfed a small aerobic bacterium. Instead of being digested, the bacterium survived and helped the host produce energy. Over millions of years, this relationship became permanent, giving you the mitochondria. So basically, the endosymbiotic theory was proposed by Lynn Margulis, who suggested that the mitochondria had evolved from free-living alpha proteobacteria. And the host cells subsequently engulfed this aerobic bacteria, fine, and then the bacteria started providing energy. Okay, and okay, so that's how this endosymbiotic theory goes. See, here you can see a larger cell swallows up the bacterium, and then the bacterium stayed alive and start help the cell to make energy. So that became the mitochondria. This is how mitochondria uh, originated. This theory is strongly supported by scientific evidence. Mitochondria have circular DNA, which is similar to bacterial DNA. They have their own ribosomes. They divide independently through binary fission. They also have a double membrane structure. All these features clearly point to a bacterial origin. So why mitochondria have their own DNA? But if mitochondria came from bacteria, why didn't they lose all their genes? Some genes are directly involved in energy production and must respond quickly to the cell's needs. Keeping these genes inside mitochondria allows faster local control. That's why mitochondria are described as semi-autonomous organelles. Fine. Mitochondria. Now let's discuss the structure of mitochondria. Mitochondria DNA has a distinct structure. It is small, circular, and double-stranded. In humans, it has around 16,569 base pairs. Each cell contains many copies of mitochondrial DNA. Unlike nuclear DNA, mitochondrial DNA lacks histones and has limited repair mechanisms, making it more vulnerable to damage. So what does mitochondrial DNA encode? Despite its small size, mitochondrial DNA is essential. It encodes 13 proteins that form the core of the energy-producing machinery. It also encodes 22 tRNAs or transfer RNAs and 2 ribosomal RNAs which are involved in protein synthesis. Okay. So without these genes, mitochondria cannot function efficiently. These are essential for, okay, so here you can see. So the central role of mitochondrial DNA is to support oxidative phosphorylation, the process that produces ATP. ATP is the energy currency of the cell. Every cellular activity depends on it. Muscle contraction, nerve impulses, active transport, and biosynthesis all require ATP. Mitochondrial DNA ensures that the proteins needed for this process are available at the right place and time. If mitochondrial DNA is damaged, ATP production decreases even if nuclear DNA is normal. That is why cells with high energy demand, such as brain, heart, and muscle cells, are the first to be affected when mitochondria fail. In short, mitochondrial DNA directly links genetic information to cellular energy supply. The central role of mitochondrial DNA is to support oxidative phosphorylation, the process that produces ATP. Fine? Yeah. So mitochondrial DNA has several unique features. It has a higher mutation rate than nuclear DNA. It lacks protective histones that wraps the DNA. It allows a special inheritance patterns called as maternal inheritance, which means that the mitochondrial DNA is passed only from the mother, which will be discussed later. These features make mitochondrial DNA very useful for evolutionary and genetic studies. It has a more compact genome. Okay, it's very small and tightly packed. It has only few essential genes. It doesn't have any introns that are non-coding strands. Fine. So they are edited with continuously. Without editing, they are read. Fine. So these are some unique features. Now moving on to one of the most fascinating properties of mitochondrial DNA is maternal inheritance. So we inherit mitochondria only from our mother. This is because sperm mitochondria are destroyed after fertilization. Both sons and daughters receive mitochondrial DNA from their mother, but only daughters pass it on. For example, if a mother carries a mitochondrial mutation, all her children will inherit it. But if a father carries the same mutation, it will not be transmitted. This inheritance patterns allows scientists to trace maternal ancestry across generations. Fine. So, what does maternal inheritance mean? So, in mitochondria, right, that is the zygote, it comes from mother. It doesn't come from father because the sperm mitochondria gets destroyed after fertilization. This sort of transmission or inheritance is called maternal inheritance. Mitochondrial DNA is linked inherently only from mother. Fine. So why does it happen? So this is important because it prevents mixing of the two different mitochondrial populations. It makes sure there's a stable energy production. Okay, so it reduces conflict between different mitochondrial DNA types. Fine. And also, another interesting fact is that mitochondrial DNA, when it's inherited only from mothers. So this allows the scientists to trace all the humans back to a single maternal ancestor, which is known as mitochondrial Eve. Now let's compare the two genomes. Nuclear DNA is large, linear, and inherited from both parents, where it controls most cellular structures and functions. It has a number of copies is to biparental. Fine. It is there is introns present and there are histone proteins which are present, which gives a protection to the DNA, and the mutation rate is low. For mitochondrial DNA, It is small, circular, and it is inherited only from the mother, maternal inheritance. Its main role is energy production. You can think of nuclear DNA as the managing the entire cell, whereas mitochondrial DNA focuses on powering it. Mitochondrial DNA also doesn't have any histone proteins to protect it, so it's highly vulnerable to mutation. So the mutation rate is much higher for mitochondrial DNA compared to nuclear. Even though mitochondria they have their own DNA, they cannot function independently. More than 99% of mitochondrial proteins are encoded by nuclear DNA. These proteins are produced in the cytoplasm and then it's imported into the mitochondria. This constant coordination between the two genomes is essential for efficient energy production. When this coordination breaks down, diseases can occur. Even though so this is okay, around it encodes most mitochondrial proteins, which is the nuclear DNA. It helps regulate mitochondrial number and the activity. It also makes sure the repair and replication is controlled. Whereas in the mitochondria, which is the energy genome, it encodes 13 energy related proteins. It sends signals when there is damage, stress signals when there is a damage to the cell. Fine. So this is how they coordinate. So the nuclear DNA is the control center, whereas the mitochondrial DNA is the energy genome. So both have to coordinate in order to have a healthy cell. So why do cells maintain two genomes? Having two genetic systems allows faster energy regulation, efficient division of labor, and greater adaptability. This dual genome system gives cells a major evolutionary advantage and made complex life possible. It makes the fast local energy. So mitochondria DNA, it allows the mitochondria to make energy proteins quickly where they are needed. Evolutionary origin, so mitochondria came from ancient bacteria. And cell survival. So it helped the two genomes. Having those two genomes helped to maintain the control the stress and energy demands. So it gave a advantage for complex life. Fine. So this is the summary. So if you sum everything up, cells have two distinct genomes, nuclear DNA and mitochondrial DNA. Mitochondrial DNA is small, circular, semi-autonomous and exists as extrachromosomal DNA. Mitochondrial DNA originated from the ancient mitochondria, whereas it has originated from ancient free-living bacteria through endosymbiosis. Nuclear DNA and mitochondrial DNA cooperate to regulate the production of energy and cellular efficiency. Mitochondrial DNA is inherited maternally, providing evolutionary advantages in speed, efficiency, and survival. To summarize part A, mitochondrial DNA is a second genome with ancient bacterial origins. It plays a central role in energy production and works closely with nuclear DNA. Together, these two genomes explain how cells evolved efficiently and evolved efficiency and complexity. In part B, we will explore how mitochondrial DNA connects to disease, aging, evolution, and forensic science. Thank you very much.